All right, everyone. It seems like there aren't as many people here this morning as usual. Did you know I was going to be talking with you about photosynthesis? Is it me or is it the subject? I wonder. But hey, that's what we're talking about today, photosynthesis. It's the flip side of what I've talked with you about the last two lectures, when I've taken you by the hand and we've walked through glycolysis, which gets us from a 6-carbon molecule called glucose, energy-rich molecule, to 2 3-carbon molecules called pyruvate. And then, in the last lecture, the second half of that process, I took you by the hand and we talked about the Krebs citric acid cycle, which takes place in the mitochondria to convert that 3-carbon molecule into carbon dioxide gas. Right? So for each glucose that we start out with, we end up with 6 new CO2 molecules that get created through the process of glycolysis, and then the citric acid cycle, or the Krebs cycle, if you prefer. Remember, an awful lot of that energy that gets captured through those processes is captured through oxidative phosphorylation, meaning that we're tying the synthesis of ATP to some movement of electrons, an electron transport chain, and setting up a concentration gradient on one side of a membrane relative to another in terms of the hydrogen ions that are present in the solution. I don't think I gave you the specifics when we talked on Thursday, but there's actually about a 1,000-fold difference in the hydrogen ion concentration from one side of that mitochondrial membrane to the other side of that mitochondrial membrane. pH 5 on the one side, high concentration of hydrogen ions, and pH 8 on the other. And when cells let those protons go down that concentration gradient, again, ATP synthase cranks this little wheel and creates one new ATP for each proton that comes through. That process has a fancy word that I don't think I mentioned, but it's a cool one. It's called chemiosmosis. Right? So in a nutshell, that synthesis of ATPs through the flow of the electrons across the membrane, through the ATP synthase gate, that business is called chemiosmosis. All right? But again, today, let's talk about the flip side. Essentially, where do those sugars, those glucose molecules come from in the first place? And the short answer to that question, or that problem, is photosynthesis. And funny thing, the whole business of photosynthesis that I'll talk with you about today is pretty much the reverse. It's the yin to the yang of what I talked with you about all of last week. So instead of starting with a 6-carbon molecule and ending up with 6 1-carbon molecules, we're going to just do that whole thing in reverse. We start with 6 1-carbon molecules, carbon dioxide gas, and we go through a number of processes to end up with a 6-carbon molecule like glucose. Pretty much, well, today really what I'm going to do is I'm going to stop when we get to 3-carbon molecules and then just tell you, you take 2 of those 3-carbon molecules and you push them together and you make a 6-carbon molecule glucose again. So that's what you're in for today. All right? So let's talk a little bit about photosynthesis, and we'll ease into it with some pretty pictures of plants and other things that do photosynthesis. This is a trick that living things have been doing on this planet pretty much since the beginning. The essence of it is we're capturing energy that's electromagnetic energy, photoelectric energy, and converting it to chemical energy. Plants are really good at pulling off that trick. They're good, and that in turn makes them what we call autotrophic organisms. They don't need an input of energy aside from sunlight to be able to drive all their chemical processes. And we can be even more specific, and we can say they're not just autotrophs, we can say they are photoautotrophs, because where they're getting their energy from that lets them be self-sufficient is from photons. That's in contrast to the other kind of autotrophs that we know about that have been studied on this planet. Those are the let me try that again. Those are the chemi-autotrophs. All right, so things that live in deep sea vents, thermal vents, for instance, use hydrogen sulfide to drive their chemical processes. Those are chemi-autotrophs. The vast, and there are things that can live just off of hydrogen sulfide gas in those deep sea vents. But the vast majority of the autotrophs on this planet are photoautotrophs. The energy that we use to drive our chemical processes in our bodies, yours and mine, comes like more than 99.99999% from our consumption of photoautotrophs. All right? We are not autotrophs. We're not photoautotrophs. We're not chemiautotrophs. There's another word for organisms like us that need to get these energy-rich compounds from something else that did the work of collecting that energy. We are heterotrophs. Okay? So organisms that need to get these energy-rich molecules from something else, it's their only way to get them. Those are heterotrophs. But organisms like plants that can synthesize their own energy-rich molecules with simply the input of sunlight without actually having to go and eat something else, those things are called autotrophs, and we can be more specific, if you like, and call them photoautotrophs. So, you probably learned this in kindergarten, but just for the record, all of this photosynthesis business is taking place in chloroplasts, right? Nobody's going to disagree with me about that, I hope. And it's those chloroplasts that cause plant leaves and even algal structures to look so green, all right? Because chloroplasts are where photosynthesis is taking place. The key pigment to the whole business is chlorophyll. I'll show you its structure in a few minutes. And that, in turn, is what gives rise to the green colors that we associate with plants. So let's take a little bit of a closer look at the structure of plant leaf. But I really want to focus our attention as much as we can today on the structure of the chloroplasts themselves. That's really where the action is for the business of photosynthesis. And, you know, how many times have I said it? Structure and function are intimately interrelated in all biological systems. Photosynthesis is still one more really strong example of how the structure of the chloroplast drives the whole process. Not just drives it, but even makes it possible, might be the best way to phrase that. So take a look. Focus your attention for a little bit on this cartoon structure of a chloroplast. You can see this is an actual microscope image. This is a nice simplified cartoon so you can see where the business is. Remember, how many times have I said this? Chloroplasts are a little bit special in that they have a double membrane-bound compartment. Like the nucleus and like the mitochondria. If you look carefully here, you'll see there's one phospholipid bilayer and another phospholipid bilayer over here. That's probably a vestige of how it is that chloroplasts came to be associated with eukaryotic cells in the first place. Again, the endosymbiont theory suggests that there had been something that was out and about photosynthesizing that got eaten at one point by a proto or a soon-to-become eukaryote. It got eaten, enveloped by that proto-eukaryote's membrane itself, and somehow persisted in a symbiotic relationship developed to give you this endosymbiotic internal organelle. So you've got the double membrane. Here's something that I haven't drawn your attention to before, right? But if you think about those three endosymbionts, the nucleus, the mitochondria, and the chloroplast, something that sets the chloroplasts apart. All three of them have that bag within a bag structure. But something that sets the chloroplasts apart is that they also have internal membrane compartments inside of that double bag, right? That's not the case for the mitochondria. It's not the case for the nucleus. But these green things here, those stacks of green things, are membranes. And they form some internal membrane-bound compartments within the internal membrane-bound compartment. So I got to throw some vocabulary out to you right now. Those green things are green because that's where the chlorophyll hangs out in a chloroplast. Chlorophyll has a long hydrophobic tail that causes it to be embedded in a membrane, right? It hangs out because of its hydrophobic tail in a membrane. I'll show you the picture of chlorophyll structure in a little bit, and I'll remind you of this. But the membrane that it hangs out in is that membrane that's shown as a green color here on that slide. And that membrane is called the thylakoid membrane. All right, so that little membrane there, that one there, this whole membrane system, that's called the thylakoid. The stuff inside, the space inside of that thylakoid membrane, very imaginative name, I'm saying sarcastically, it's called the thylakoid space. Once you get past the weird word thylakoid space is a gimme, right? So try to remember thylakoid, and then the space inside of the thylakoid is called the thylakoid space. The space outside of the thylakoid, so this stuff over here, outside the thylakoid, but still inside the inner membrane of the chloroplast, that's called the stroma, S-T-R-O-M-A. Okay? So you can't tell a player without a game card here. There are a couple of important players for the purposes of what I'll be talking with you about today. There's the stroma, the thylakoid, and the thylakoid space. The thylakoid is essentially that stack of membranes that is where chlorophyll hangs out. All right, so this is the structure, and as I talk you through today's lecture, I'll be telling you about how it is this structure in turn supports and lends itself to the whole business of photosynthesis. Before we do that, let's get some of the gross chemistry out of the way. Gross in all senses of the word, right? I'm going to talk to you about the whole package here of the chemistry. The overall reaction, in very simple terms, is what I've got written for you on the outline here. You take carbon dioxide gas, you combine it with water to make, check that out, you could call this a carbohydrate. It conforms to that formula we've talked about for a carbohydrate. Carbon and then water, H2O, and oxygen gas. So substrates for the reaction are carbon dioxide and water. Products of the reaction are carbohydrates, and this is effectively a waste product for plants, oxygen gas. Let's look at it in a little bit more detail. It may not mean that much to you guys today, but for the sake of history and acknowledging all that's transpired here, let me just mention one thing. It turns out that in figuring out how it is that the chemistry of photosynthesis worked out, it was a big deal to figure out for the first time that the oxygens that end up being part of the carbohydrates are coming from carbon dioxide gas, not coming from the water. All right, that was a watershed moment. Everybody up until that point had been assuming that the oxygens were coming from the water. Turns out the oxygens come, and the carbons, I hope, obviously, come from the carbon dioxide gas. The way this process all plays out is this. You start with 6 carbon dioxides and 12 waters. You end up making one 6-carbon molecule. So again, that bookkeeping trick, the accounting trick that I'd suggested you keep an eye on for what was going on with the citric acid cycle is going to be important here again today as well. 6 1-carbon molecules end up giving rise to one 6-carbon molecule. You have the 12 waters that come into play here. Along the way, 6 new water molecules actually get made. So there's a net consumption of 6 waters. You started with 12. You end up with 6. You've consumed 6 along the way. And you make 6 oxygen gas molecules. So that's looking at the chemistry of what's playing out here in a very high-level sort of way. To think for a moment about the energetics, you start off with things that are low-energy molecules, right? Carbon dioxide gas, if you picture that structure, those are polar covalent bonds. Those mean low energy bonds. If you look at the structure of water, H2O, again, polar covalent bonds. They're pretty much the definition, the epitome, the poster child for low energy bonds. So you're starting out with things that have low energy covalent bonds. They're polar covalent bonds. And you end up with stuff, these two things in particular, that have a lot of energy associated with them. Nonpolar covalent bonds here, nonpolar covalent bonds here, or at least a bunch of them, right? And so that's the name of the game. We're trying to we're trying to capture energy in polar or nonpolar covalent bonds. And so the whole process is pretty much about how do you get some energy to be able to drive these chemical reactions so that you end up with this product in particular. Well, the answer to that question is photosynthesis. That's how you capture that energy. And photosynthesis takes place in two distinct phases. There's a set of reactions that are called the light reactions, and a set of reactions that are called the dark reactions. It shouldn't shock you to hear that the light reactions require light. All right, that's what makes them the light reaction. It's not that they don't weigh as much, right? It's that these are the reactions that are driven by photons, by the absorption of energy associated with photons. The dark reactions can take place all the time, but they're not dependent. You don't have to have photons present at the time for the dark reactions to take place. And from a sort of strategic or a tactical kind of way of thinking about what's happening here, the light reactions are where you're making ATPs and NADPH, right? That's where you're capturing the photoelectric energy in those high energy bonds of ATP and the high energy electron of NADPH. The dark reactions are where you take those energy-rich molecules, ATP and NADPH, and you use them to synthesize those carbohydrates. Okay? So that's the light reactions. And the dark reactions, the slide right now is showing you that the absorption of light can let you drive the light reactions. And then it's those high energy molecules that get made that let you crank the wheel of something called the Calvin cycle. Pretty much the opposite of the Krebs citric acid cycle. But you're taking these high energy molecules now to drive this cycle, and with each crank of that wheel, with each crank of the Calvin cycle wheel, you make another carbohydrate, a sugar. All right? Excuse me. So that's it. That's it sort of from a high-level perspective in terms of what the chemistry here is about. Let me remind you, this is another one of these redox processes. You don't always get to see the charge moving, but pretty much each step along the way here, the key thing to keep your eye on is the movement of a high-energy electron as it's getting passed along an electron transport chain, as it's being passed from NADPH onto something else. All of these chemical reactions fit into that category of redox processes that we've talked about for the last week now. So I've said it a bunch of times here today. Big part of what has to happen is we want to convert photoelectric energy into chemical energy. Chemical energy is the energy associated with covalent bonds. In Gibbs free energy terms, it's the enthalpy, right? That's the kind of energy that we want. That's the kind of energy that we need. It's something we can hold in our hands, and we can physically, and physically is the key word here, physically link to other chemical reactions to hook up energetically unfavorable reactions to favorable reactions. And again, living things do that trick to help certain chemical reactions take place. So the trick is, how do you get that chemical energy when you're starting with photoelectric energy? Let's talk for just a little bit about the energy associated with photons so you get a better feel for what kind of energy we're talking about and how it differs from that chemical energy. At this point in the class, I think you're all really comfortable with the chemical energy. Polar bonds, not so much energy, nonpolar bonds, lots of energy associated with them. Well, photons have energy associated with them. No big surprise associated with that statement, I hope. But photons are kind of interesting in the sense that they behave both as particles and as waves. Okay, they have dual characteristics. They're equally best considered as a particle and as a wave, the particle being the photon. But the photon is moving in a wave. The closer together the two peaks of a wave are, the crests of a wave, the more energy a photon has associated with it. So the length of a wave, the distance from one crest to the next crest, or if you prefer, from one trough to the next trough, right? The distance of that distance is called the wavelength. It's the length of the wave. The shorter the wavelength, the more energy a photon has associated with it. In this slide gives you a whole spectrum, and that is the perfect use of the word spectrum. This is what spectrum initially referred to. You can start out with things with crazy small wavelengths, okay, like gamma rays, 10 to the negative fifth nanometer, right? Billionths of a meter from the crest of one wave to the crest of the next wave in this type of light. You can start out with things with ridiculously small wavelengths that have enormous amounts of energy associated with them. Each of those photons packs a real punch because it has such a short wavelength. But at the other end of this spectrum, you can see there are things like radio waves. And whether we like it or not, you and I are awash in radio waves right now, and we don't notice or care, right? But there's radio signals coming from each of our phones. There's radio signals coming from, local radio stations. I mean, we're just being washed constantly by radio waves. But it doesn't bother us because their wavelengths are so large. They can be kilometers long, miles long, the distance from one crest to the next crest. Those long wavelengths correspond to very small amounts of energy. Those photons, when we absorb a radio wave, we barely notice it. It's got such low energy, it doesn't make us warm, it doesn't make us, we don't notice it at all. And somewhere in between is the visible spectrum of light, right? These are the wavelengths of photons that you and I can perceive with our eyes, the visible wavelengths of light. It's pretty much smack in the middle in terms of this spectrum. At the short end, the low wavelength, the smaller wavelengths, like around 380 nanometers, that's where we see the color purple. Photons that have that wavelength associated with them, when those hit our eyeballs, we think and perceive purple. When we have photons with longer wavelengths, like 750 nanometers, that's when we're seeing the color red. When those photons hit our eyeballs, we perceive red. And of course, there's everything in between. You go from the purples to the blues, the greens, the yellows, the oranges, ultimately to the reds. Again, the shorter the wavelength, the more the energy associated with those photons. Chlorophyll, the star of photosynthesis in terms of absorbing photons, chlorophyll absorbs light best in two different ranges within the visible spectrum. And chlorophyll does not absorb light that's outside of the visible spectrum, at least not in a way that we worry about too much. It's at a very low level. Chlorophyll does a great job of absorbing photons with these wavelengths, the purples through the blues, and it does a really good job of absorbing photons with these wavelengths, the yellows through the reds. So chlorophyll is all about a photon comes along and chlorophyll latches onto it, it absorbs it, it takes its energy to itself, and then it's that gets used to drive the light reactions of photosynthesis that we'll talk about in a little bit. But I'm sure you've noticed that I haven't said that chlorophyll absorbs this wavelength of light. Okay? Chlorophyll, sort of counterintuitively, we always think of chlorophyll as being green. At a first pass, you might think chlorophyll is really good at absorbing green light. It's actually just the opposite. Plant leaves look green because the chlorophyll is absorbing all the rest of white light. All of those wavelengths of light together are white light, like what we have in this room right now, coming from the light bulbs. That's got some photons that are red, some photons that are yellow, some photons that are green, purple, and blue, right? Got the whole spectrum in white light. But what plant leaves are doing is they're selectively pulling out the photons that are purple and blue, and yellow and orange and red, and leaving behind, not interacting with the photons that have green wavelengths associated with them. That's why chlorophyll looks green. It's because it's absorbed all the rest of the photons, and the only things left behind are the green ones. So again, somewhat counterintuitive, I think. At least it has been for me. And that ultimately causes, this picture sums up in a cartoon what it is I've just been trying to tell you. Again, when you start with light coming from the sun, that sunlight is white. It's got photons of all imaginable wavelengths, not just in the visible spectrum, but outside of it as well. But let's focus on the visible ones because that's where chlorophyll is going to be absorbing photons. You start with white light from the sun, and when those photons interact with chlorophyll, or any matter for that matter, one of three things happens. When a photon interacts with matter, it can get absorbed, it can get reflected, or it can get transmitted. Chlorophyll does a great job of absorbing the photons in the red part of the spectrum and in the purple and blue part of the spectrum. But for the green part of the spectrum, it does both reflect and transmit. So those photons are the ones that get away to go off and hit our eyeballs. The red ones, the yellows, the blues, and the purples get absorbed. Their energy gets captured by the chlorophyll molecule and can be used then to drive the light reactions of photosynthesis. I've promised this to you a couple times here today. There's the chemical structure of chlorophyll for you, for the record, so you know what it looks like. Let me point out a couple of the highlights. I'll be a tour guide for you here. Here are a couple of the interesting or more important features of a chlorophyll molecule. Let's start at the bottom, right? I've already mentioned this to you, but chlorophyll has a long hydrocarbon tail. That's code for hydrophobic hydrocarbon, right? Remember that from our earlier discussions about these types of structures. There is nothing going on here but carbon to carbon and carbon to hydrogen bonds across this whole tail. It is the very definition of hydrophobic. It wants nothing to do with water. It is really comfortable, really happy if that is embedded in a lipid bilayer. Which one? The thylakoid membrane's lipid bilayer. All right, so that's what's anchoring chlorophyll in the thylakoid membrane's membrane. But also attached to this hydrophobic anchor is something that's called a porphyrin ring. Lots of pigments have porphyrin rings associated with them. This particular pigment is chlorophyll. A lot of things that absorb light have these porphyrin rings. Here again, we're talking about chlorophyll. This ring structure, pretty complicated structure, right? Don't worry about memorizing it, but that's what the porphyrin ring structure of chlorophyll looks like. Notice in the very center of the porphyrin ring is a magnesium. You don't see magnesium showing up too often in biology classes, but it's a key player for chlorophyll. It is actually that magnesium that contains the electron that gets excited by the absorption of photons. So a photon interacts with the matter associated with this porphyrin ring, transfers that energy ultimately to the magnesium. And what happens within the magnesium atom itself is you take an electron from a low energy state and you kick it up to a higher energy state. In fairly simple terms, instead of being in one of the lower level orbitals, it goes to a higher level orbital. It goes further out from the nucleus than it was when it started. That's where the high energy electrons start, and that's also where a high energy electron will need to be replaced to be able to do this process again. All right. So enough about electromagnetic energy, and maybe even enough about the photooxidation of chlorophyll. Let's talk about how it is that these high energy electrons actually get put to use. I hesitate to show you this picture just because maybe it's too much at this point in the lecture, but here's sort of like the big picture of what's going on. They also have given you a lot of details here. But the essence of what's happening is this. Photons coming from the sun get absorbed by a light harvesting complex within thylakoid membranes. That light harvesting complex is a bunch of chlorophyll molecules hanging out together. Right? Each of the green dots is supposed to make you think of a chlorophyll molecule. So a photon gets absorbed by a chlorophyll. The chlorophyll molecule gets its magnesium electron excited. That excited electron can bop around from one chlorophyll to another, right? From one magnesium to another, until ultimately it gets dumped off onto some special chlorophylls, all right, that are part of this light harvesting complex. And it's there that these special chlorophylls shunt the electron off to the reaction center complex. Right? And so these are pigment molecules. They're typically chlorophyll, but there are also other pigments, like the things that cause leaves to be so pretty at this time of the year as the chlorophylls being reclaimed by the plants before the leaves get shed. A lot of those pigments are left behind. You get to see them. Those are in play as well, but the big player is chlorophyll. And here, in a little more detail, is what's going on with those photocenters with the absorption of the energy. So this diagram actually has some numbers associated with it to help you keep track of the sequence with which things happen. It all starts when an electron in a magnesium from a chlorophyll gets excited by the absorption of a photon. That excited electron bops around until eventually it makes it to photosystem 2's special chlorophylls that then take that high energy electron and deliver it to a primary electron acceptor, and that electron gets replaced by a low energy electron that's coming off of a water molecule, and as a result you start to make some oxygen gas. Okay? So a number of things have happened here. The big picture: photon excites an electron. That excited electron gets passed off to this receptor. The receptor puts it off into the primary receptor, and the electron in the chlorophyll gets replaced with an electron that comes from a water molecule as you're making oxygen gas. Here's where the action starts to happen for real. We've got an excited electron with a lot of energy associated with it. And that excited electron goes through an electron transport chain. This is the second time in this class I've talked with you about an electron transport chain. Remember, that was a big deal in the context of oxidative phosphorylation. That's where you were getting most of your ATPs from the business of converting pyruvate to carbon dioxide in the citric acid cycle. We've got here again another electron transport chain. On Thursday of last week, I suggested it was sort of like a bucket brigade. You have a high energy electron, you pass it off to the next guy in line, there's a little less energy. You pass it off to the next guy in line, a little less energy. Each pass along the way. It's energetically favorable. You're going down an energy gradient. Along the way, as this high energy electron gets passed from a cytochrome here to a cytochrome here, along the way, guess what happens? Just like was the case for the citric acid cycle and with oxidative phosphorylation, a proton gradient gets set up. It's the very same general idea. We set up a proton gradient, pH 5 on one side of a membrane, pH 8 on the other side of a membrane, and an ATP synthase sits in that membrane, allows protons to go across that gradient, down that gradient, and along the way turns a crank and synthesizes ATP. So for each of these electrons that gets excited by a photon and pawned off here to P680, these special chlorophyll molecules, for each photon that happens to, a proton gets pumped across a membrane, and for each proton that gets pumped across a membrane, we've gotten an ATP. We can make one ATP that we didn't have before. Sweet deal, right? We're plus one ATP for this whole process, again, because of oxidative phosphorylation. But that's not the end of the story. You can tell this is photosystem 2. Since there's a photosystem 2, it kind of suggests there's a photosystem 1. If you've been thinking along those lines, you're right, because sure enough, there is another photosystem that can accept those high-energy electrons, because they're still pretty high energy, even after being used to set up that proton gradient. Photosystem 1 can take those electrons and then run them through another process that allows you to directly make NADPH. After another electron transport chain takes those high-energy electrons, at the end of the day, you make, for every electron that runs through that electron transport chain, you make an NADPH, whereas you had started with just an NADP and a hydrogen. At the end of this whole process, we can say we've made, from one excited electron, an ATP and an NADPH. Not bad for a day's work. It's possible for you to make the NADPH directly by having a chlorophyll interact here. You don't get the ATP if you do it that way, if you start with photosystem 1 as opposed to photosystem 2. If that was a cartoon, the slide on the screen right now is an Uber cartoon, right? But this is another way to think a little bit about what's happening here. You get a photon on the left there that gets excited, going up, right? You're investing energy in it. That electron gets handed off to an electron transport chain. Along the way, you turn a crank by setting up that proton gradient, and that crank makes you an ATP. Photosystem 1 then accepts that excited electron, sometimes juices it up with a little bit more energy still, and gets you to a point where you can run it through another electron transport chain that ultimately delivers an NADPH. Here's a detail. It's a detail, and I'll probably end up asking you a question or two about this on the second test for the class, just to make sure you're paying attention to this level of detail. In other classes, it's going to be important for you to realize the flow in which the electrons are the path, the flow that they're taking through this process. There's one of two choices, right? The electrons can just cycle in a little circle like you're seeing here, where the excited electron comes in, runs through photosystem 1, and instead of getting passed off to give rise to the NADPH, can go back through that process again. You can just keep recycling that electron and make just ATPs. I used the word recycle there a couple times. It's a good word to use. What this is describing is what's known as cyclic electron flow. And that's in contrast to this non-cyclic flow. The electron starts here, passes along through here, and ends up getting dumped off onto this NADPH. The electrons for the non-cyclic flow have to be replaced by water molecule electrons. The electrons for the cyclic flow are just coming back in a loop themselves. A chlorophyll electron is replacing another chlorophyll electron, and you're just running it through a cycle and juicing it up with the photons that are being absorbed in the process. So we've got two photosystems. We've got some flow associated with those electrons. Let's talk now about the details of the synthesis of that ATP, the oxidative phosphorylation. Again, in contrast to substrate level phosphorylation, this is oxidative phosphorylation. So what's going on here is this, is you've got yourself a thylakoid membrane. Embedded in the thylakoid membrane, right, this is the membrane inside of the double membrane of the chloroplast. Embedded in the thylakoid membrane are chlorophyll molecules. They're absorbing photons all the time. When a photon gets absorbed, it passes around from one chlorophyll to another until ultimately it gets into photosystem 2's receptor and then gets passed off through an electron transport chain. As the electrons go from high energy to lower energy through that electron transport chain, hydrogen ions get moved from the stroma, there it is, to the cleverly named thylakoid space. So you have a low concentration of hydrogen ions over here, pH 8, if you want to put a number to it, and a high concentration of protons here, pH 5, if you want to put a number to it. Notice the shading. They're trying to help you out here with the colors, and you'll see a slide in another few minutes that this shading continues with. Where there's a lot of protons, they color it darker gray. Where there's not as many protons, they show a lighter gray. Okay? That proton gradient gets relieved, right? The deal we talked about, remember the deal we talked about on Thursday? Nature, let's cut a deal here. I know it freaks you out. You don't like that there's so many protons here. The concentration's irritating. The charge is irritating. I'm going to help you out. I'm going to let some of those protons get across the membrane. All I'm going to ask in return is that you drive the crank that lets my ATP synthase make an ATP for each proton that I let go across the membrane. Right? That's the deal that was playing out within the mitochondria. The central, the protein that does it is ATP synthase. Guess what? It's the same protein, ATP synthase, that chloroplasts are using. I suggested to you on Thursday that ATP synthase is probably one of the very first proteins that living things came to use. They've been using it ever since. It hasn't changed very much. We see the same protein being used by both mitochondria and chloroplasts for this process. Again, it's one of the first proteins that's probably been available for living things on this planet to use. But it's the same deal. We let protons go through. As they pass through to go down that concentration gradient, ATPs are being made. So here's a graphic that shows you what I had told you in the earlier slide. You set up a proton gradient, and you use that proton gradient to make ATPs. At the same time, these high-energy electrons can work their way through to ultimately get passed off from photosystem 2 through an electron transport chain and delivered to photosystem 1, and then photosystem 1 runs them through another electron transport chain that at the end of the line has an enzyme called NAD+ reductase that takes hydrogens and fuses them or attaches them to NAD+ to give you NADPH. NADP is very similar to NAD, right? NAD was the molecule we were talking about, or I was telling you about in the context of the citric acid cycle. NADP differs from NAD, guess how? It's got a phosphate group attached to it. It's the only difference, subtle, but nonetheless a difference that gets recorded when you're talking about what's going on here with chloroplasts. You're making not NADH, the energy-rich molecule from the citric acid cycle. Instead, you're making NADPH. That NADPH and NADH have equivalent amounts of energy associated with them. That's an energy-rich molecule. All right, I threatened that I'd show you a slide like this, right, where the shading was important. If you can, just look at what's going on the right side of the screen. In a little bit, we'll talk about the left. I'll tell you on the left is what's happening in mitochondria. What's happening on the right are chloroplasts. This is what I've been talking with you about so far today. Look at that pretty chloroplast. Isn't it a nice chloroplast? It's got its double membrane there. It looks like a chloroplast, right? You've got your stroma here, light shade of gray, meaning there's not a lot of protons there, pH 8, right? And you've got your thylakoid membrane with thylakoid spaces with darker shading, pH 5, higher concentration of protons here than over here. Across that membrane is a gradient. We can zoom in and take a look at it. And what's happening is inside the thylakoid space, you've got a lot of hydrogens, and you're letting them go to the stroma, and along the way, you make ATP. Where does that ATP end up? It's an interesting question, at least to me. It's kind of an interesting question. Where are the new baby ATPs showing up? You know where the new baby ATPs are? They're in the stroma. The orientation of the ATP synthase, the way that that crank is running, the way that it's poking and sitting in the membrane, the new ATPs are being made in the stroma, not the thylakoid space. They're being made in the stroma. All right? That's what's happening with photosynthesis. An electron transport chain is involved, right, to set up that proton gradient in the first place. At the end of the day, you're making shiny new ATPs, and they're coming out in the stroma, this part of the chloroplast. Not that hard to get them from here to outside the chloroplast into the cell where they can be put to use to make things like sugars in the dark reactions of photosynthesis. Or better, really, the dark reactions of photosynthesis take place in the stroma. That's right where they need to be. Okay? Compare that to what's been going on in the mitochondria. A lot of the players are the same. Remember, electron transport chain, a lot of the same proteins and complexes are involved in the mitochondria as what we see in the chloroplast. The ATP synthase is the very same ATP synthase in the mitochondria and the chloroplast. So a lot of the players are the same, but where they're playing is different. For mitochondria, what's going on is you're setting up a proton gradient where it's the mitochondria intermembrane space, the space between the outer membrane of the mitochondria and the inner membrane. That's where you have your pH 5. And your pH 8 is in the very center part of the mitochondria. When you're making shiny new ATPs in a mitochondria, where you're making them is in the inner mitochondrial matrix, this innermost space. That's where the shiny new ATPs are getting made. It's a little different than what's happening here with the chloroplast. Right? It's kind of like the ATP synthases and the electron transport chains are pointing in opposite directions relative to the membrane in the mitochondria relative to the chloroplast. That's how it's set up, right? Works fine either way, apparently. For mitochondria, that's the way things are working. For chloroplasts, it's working in exactly the opposite orientation. Okay, so those are the light reactions of photosynthesis. Remember, they're driven by the absorption of photons. They can only take place in the presence of light. That's why we call them the light reactions. There's another set of reactions that take place with photosynthesis, the dark reactions. So we're halfway through, right? We've done the light reactions. Let's talk a little bit about the dark reactions. The dark reactions are the chemical reactions that ultimately are going to make us some carbohydrates. Because if you think about it, the light reactions haven't made us any carbohydrates. They've just made us ATP and NADPH. Swell. Nice to have those energy-rich molecules, but you want that $100 bill in your purse or your wallet, you gotta lock it in by making glucose molecules. So the light reactions have made us out of nothing ATPs and NADPHs. To really lock those things in and to keep them for a rainy day, literally, when there may not be as much sunshine around, right? To lock them in, we need to lock them in as carbohydrates. And how that happens is ultimately with the dark reactions of photosynthesis. The dark reactions of photosynthesis happen exclusively in the stroma of chloroplasts. So the enzymes that are catalyzing these reactions are in the stroma. The stroma, remember, is also where those shiny new ATPs and NADPHs are. So pretty good design system so far. You don't have to move those ATPs or NADPHs anywhere. They're right where the enzymes are that are going to catalyze the dark reactions of photosynthesis. So we got some chemical reactions that need to take place. The big player in the dark reactions of photosynthesis is an enzyme called ribulose bisphosphate carboxylase. Between friends, since 1979, known as Rubisco. Okay? I was just reading this morning. I didn't know the history of the term Rubisco. That's why I happen to remember 1979. Before 1979, everybody just called it ribulose bisphosphate carboxylase. But in '79, the guy who had worked a lot of this stuff out, Calvin, was retiring. And in a tribute to him, they had like a party or roast, right? And some other scientist, as a tribute to him, said, you know what, instead of calling it ribulose bisphosphate carboxylase, from now on, I think we should call it Rubisco. Ribulose, RU, bisphosphate, the bis in Rubisco, and co is sort of chemist slang for carbon dioxide or carbon monoxide, really. But so he comes up with Rubisco. He suggested it as a joke. And he suggested it as a joke because the scientist who was getting roasted had also been suggesting that this protein, Rubisco, could be used to make a protein supplement that would be useful for baby formulas and mass-produced all over the world so that people could have that protein as a protein supplement. And he did it deliberately because he thought Rubisco sounded sufficiently close to Nabisco, the food processing company, that he wanted to sort of marry those names together. Regardless of that strange history, the name Rubisco has stuck. Rubisco's a big deal, okay? By most people's reckoning, Rubisco is the single most abundant protein on this planet. All right? Probably the most abundant protein in our bodies is hemoglobin. Maybe there are a couple others, myosin, actin, that might give hemoglobin a run for the money. But on the planet overall, Rubisco, a big deal. There's lots and lots of Rubisco on this planet. By a lot of people's reckonings, the single most abundant protein on the planet. Why is Rubisco the most abundant protein on the planet is an interesting question in its own right. Two reasons. One is you can't do the dark reactions of photosynthesis without Rubisco. Right? It's central to the whole business. Anything that's doing photosynthesis, there's lots and lots of biomass doing photosynthesis. Anything doing photosynthesis has to have Rubisco to pull it off. And the second reason, Rubisco isn't that well designed an enzyme. It's got some issues. One of the issues that Rubisco has is it's slow. Its turnover rate. Remember I was talking with you guys a while back trying to impress you with how fast enzymes were? 100,000 products to substrates per second for catalase, that kind of thing. Rubisco is way at the other end of the spectrum. It's a real slow enzyme. Its turnover rate, depending on the organism that you've gotten it from, ranges from 3 to 10 per second. So you know that some enzymes can really crank out products, hundreds of thousands of products per second, millions for some per second. Rubisco takes its sweet old time compared to other enzymes. 3 to 10 for a turnover per second, pretty fast by our standards as big, oafish, clumsy humans. But at a molecular catalytic sort of level, Rubisco takes its sweet old time. Okay, and so for those two reasons, the fact that everything that does photosynthesis needs Rubisco, and the fact that you need to have a lot of Rubisco, because Rubisco molecules by themselves don't work that fast, that contributes to making Rubisco in the running a serious contender for the most abundant protein on this planet. So, one reason to remember Rubisco, all right? The other reason is because it plays an awfully important role. So I'm trying to telegraph to you here. There's a bunch of enzyme names involved with the Calvin cycle that we'll be talking about here today. There's really only one of them that I think you want to be able to trot out when you're having a conversation with somebody like Don Cipollini next semester, and he's talking with you about plant physiology, right? Rubisco. Man, if you don't know Rubisco, he's going to be very disappointed in you and in me. So that's one enzyme, perhaps, that you want to keep in your memory. So what's Rubisco do that makes it such a big deal? What makes it a big deal is Rubisco is the enzyme that is solely responsible for carbon fixation. What we mean when we say carbon fixation is you've got carbon in a gas, carbon dioxide, bopping around, doing its own thing, devil may care, whatever, having a good old time, and then Rubisco comes along and fixes it. It ties it down into a solid. It takes it out of that unfixed, floating around gaseous state, and it anchors it in to by covalent bonds to other carbon atoms to make a carbohydrate. Rubisco, if there's nothing else you remember about Rubisco, the trivia about how it's a slow enzyme and it's so abundant and all that, if you have to remember one thing about Rubisco, it's the thing that does carbon fixation. And that's the way people describe it. The carbon gets fixed into a solid form as opposed to a gaseous form. What Rubisco does is it takes two substrates, carbon dioxide gas and another substrate called ribulose bisphosphate. Remember I said it was ribulose bisphosphate carboxylase? Well, the name of the enzyme comes from ribulose bisphosphate, carbon dioxide. It's a carboxylase. That's the co part, right? You take these two substrates and you use them to make a 6-carbon molecule, a carbohydrate. So you start with a 5-carbon molecule. If you look at this 5-carbon molecule, just in the very sketchy way that it's been drawn out in this diagram, I hope what you're seeing here is those are each supposed to be carbons, and the P's are supposed to be phosphates. I hope you guys are so savvy with looking at these kinds of structures at this point that you look at ribulose bisphosphate and you say, that's got all the hallmarks of an energy-rich molecule, right? Just at a glance, that's looking pretty energy-rich, right? It's a sugar, it's got phosphates on it. That looks like it's something that's primed and ready to go. And what's happening is you're taking that energy-rich 5-carbon molecule, ribulose bisphosphate, bis because there's 2 phosphates, one on each end of the molecule, and you're using it to, some of its energy to bring together and force it into a union with that 1-carbon molecule to make a very transient 6-carbon molecule that falls apart into 2 3-carbon molecules called 3-phosphoglycerate. Carbon has been fixed. It had been a gas. It's now a solid. The solid falls apart to give us this particular sugar, 3-phosphoglycerate, because the phosphate's on the 3rd carbon in this molecule. This molecule gets ultimately converted into glyceraldehyde 3-phosphate. And you can take 2 glyceraldehyde 3-phosphates, smush them together, and make glucose out of them. How do you manage to make glyceraldehyde 3-phosphate? Well, you manage to make it by investing a lot of ATPs and a lot of NADPHs. Where do those ATPs and NADPHs come from? They come from the light reactions of photosynthesis. So you can't do this part, the dark reactions of photosynthesis, unless you have the light reactions making those energy-rich ATPs and NADPHs. If you have to remember a couple of words along the way here, I'm not too worried about if you know what 3-phosphoglycerate is or 1,3-bisphosphoglycerate. Glyceraldehyde 3-phosphate, that's a big player because it's the thing that gets shunted off to make glucose. Ribulose bisphosphate is another big player because it, with carbon dioxide, plays a pretty important role in carbon fixation, and Rubisco is another big player. Well, at this point in the process, we need more ribulose bisphosphate, and that can be made through a little more energy investment, fusing together these 3 carbon molecules to give you 5 carbon molecules. You need some ATPs to drive that process. Where do those ATPs come from? They come from the light reactions of photosynthesis. Now, the elephant in the room, the thing that's probably disturbing you and making you worried that you're missing something, is what's with the 3? What's with the 3 over here? What's with that 6? What's with the 6 over here? What's happening with the Calvin cycle, and that's what we're talking about here, again, taking place inside the stroma. What's happening with the Calvin cycle is you're cranking this wheel one time for every carbon dioxide that comes in. For every carbon that gets fixed, you crank the wheel one time. But the end product here, the thing that you really like, that's really useful about this cycle, is something that's got 3 carbons, glyceraldehyde 3-phosphate. So one turn of the wheel isn't enough to give you put in 1 carbon from your accounting or bookkeeping perspective. That's how you're going to end up with something with 3 carbons at the end of the process. What you actually have to do is realize that there's a population of molecules that are moving through this cycle at any given point in time. Every 3 times you crank the wheel, you effectively get 1 more glyceraldehyde 3-phosphate. And so what's taken place, again, the numbers work out if you pay attention to what's happening here. You've got 3 carbon dioxides that come in. Let's think about them all coming through together in one fell swoop, one pipeline, that are hooked up with 3 ribulose bisphosphates. So 3 times 5, 5 carbons here, 1 carbon there. 3 times 5 is 15. 3 times 1 is 3. 15 plus 3 is 18. You get 3 6-carbon molecules, total of 18 carbons at this point. We had 15 plus 3 gives us 18, and the 18 are still here. Those 3 6-carbon molecules break up to give us 6 3-carbon molecules. 6 times 3, 18. Still have 6 3-carbon molecules here. Still have 6 3-carbon molecules here. But we're going to let one of those fall off of the cycle. We're going to shunt it off, and we're going to end up now with 5 3-carbon molecules and 1 3-carbon molecule. So 5 times 3 is 15. 1 times 3 is 3. 15 plus 3 is 18. We've still got the 18 carbons in play. 3 of them got shunted off, and now we've got 5 3-carbon molecules that we dress up to give rise to 3 5-carbon molecules called ribulose bisphosphate. 15 carbons here, 15 carbons here. We're going to add 3 more to the mix. And so you can imagine how figuring this out may have been a challenge, right? Hats off to Calvin, who came up with working out how all this plays out. You have to realize it's a cycle, and there's actually a population of molecules that are going through. And what ends up happening is for each 3 turns of that cycle, you fix 3 carbons, and you've made 1 new 3-carbon molecule that you hadn't had at the start. Those 3 carbon molecules, the glyceraldehyde 3-phosphates, are gold because that glyceraldehyde 3-phosphate, as they call it here, is a sugar. It's a carbohydrate. It's got a lot of energy associated with it. For goodness sakes, it even has a phosphate dangling off of it, another one of the hallmarks of an energy-rich molecule. And it's a hop, skip, and a jump to combine two glyceraldehyde-3 phosphates into one glucose molecule. You lose your phosphates, you get your 2 3-carbon molecules together, and you make it a 6-carbon molecule glucose. And now we're in a position where you can start the whole process again, but in the other direction, where you run glucose through glycolysis and the citric acid cycle to make ATPs. All right? This is how we synthesize glucose. Last week, we talked about nothing but how it is you extract the energy from glucose to make the ATPs and the NADHs that you want to use to drive other reactions. The light reactions make ATP and NADH, or NADPH. That energy gets locked in a more stable, less transient form when we lock it into glucose to make things like starches and carbohydrates. And then when we want to extract that energy, we take our energy-rich molecule to the bank, and the bank runs through glycolysis and the citric acid cycle to give us back ATPs and NADHs. So those are the dark reactions of photosynthesis. One more thing. I've said a few disparaging things about Rubisco this morning. I've suggested to you that it's slow. Let me tell you what its worst rap is. Right? Rubisco, a lot of people look at Rubisco and they say, this is an instance where evolution didn't quite get it right, and not lacking any hubris, thinking there are researchers who are saying, I can design a better enzyme than Rubisco, something that does a better job than Rubisco. And so there's federally funded research where people are actually tinkering with the Rubisco enzyme, its amino acid sequences, thinking that they're going to be able to design something better. What's wrong with Rubisco? Well, I've already told you it's slow. It would be swell if it could be a little bit faster. But even worse, much worse, Rubisco has a really hard time distinguishing between carbon dioxide, one of its substrates, and oxygen gas. That's a real problem for plants and for Rubisco in particular. Remember what Rubisco does with carbon dioxide gas. It fixes the carbon. How does it fix it? It attaches that carbon by covalent bond to ribulose bisphosphate to make that transient thing that ultimately breaks apart to the 3-carbon molecule and you start your Calvin cycle. What Rubisco does, if it mistakes oxygen gas for carbon dioxide, is it takes oxygen gas and attaches it to ribulose bisphosphate. And the end product of that is not very useful to cells. The only way that that thing is useful to cells is if the cell invests in ATP in a 6-step process to convert it back to ribulose bisphosphate, to get the oxygen off of it, all right, and get you back to ribulose bisphosphate, which you can use again by Rubisco, and hopefully this time not make the mistake of fixing oxygen and instead fixing carbon dioxide. Rubisco has a hard time distinguishing between carbon dioxide and oxygen. They're about the same size. They have oxygens on both ends. They fit into the active site pretty comfortably for both of them. Rubisco makes this mistake a lot. The higher the concentration of oxygen relative to carbon dioxide, the easier it is for Rubisco to make that mistake. It's easy to imagine, right? Picture the active site. You got 10 carbon dioxides bopping around and 1 oxygen bopping around. Yeah, if there's equal preference by Rubisco for oxygen and carbon dioxide, if you got a higher concentration of carbon dioxide, you won't be in such bad shape. But Oxygen gas in our atmosphere today is about 500 times more abundant than carbon dioxide gas. The planet we live on right now has a lot more oxygen gas in its atmosphere than carbon dioxide. Rubisco and plants get a bit of a break because oxygen gas, because it's got nothing but non-polar covalent bonds, is not as soluble in water, like what's inside of cells, as carbon dioxide is. But even still, within chloroplasts, carbon dioxide is only present at about 1/25 the concentration as oxygen gas. And so this is a problem. What I've described to you here, this business of Rubisco mistakenly using oxygen gas instead of carbon dioxide, has a name. It's called photorespiration. It's a wasteful process. It costs cells opportunity. There's ribulose bisphosphate that's tied up now doing something that's not very useful to a cell until it can get fixed. It's wasteful in terms of the opportunity cost. It's also wasteful in the fact that it takes an investment of energy, ATP, to undo the mistake, to fix the mistake that you made in the first place. So there's nothing good about photorespiration from the perspective of a cell, but it's happening all the time. How could this have come to be? We usually look to nature for solving problems in clever ways and ways that are very logical. You look at it, oh, that's just the way I would have designed it. Rubisco and its problem with distinguishing between carbon dioxide and oxygen, that's not the way any engineer would have sat down and designed Rubisco. How did this happen? The speculation, the thinking is that it happened because when Rubisco was getting worked out, right? When evolution was creating and tinkering and messing around with setting up a protein that could do carbon fixation, when Rubisco was getting designed and tweaked and optimized, the planet was a lot different. There was a lot more carbon dioxide in the atmosphere than oxygen. Oxygen is only built up with the success of plants spewing out oxygen as a waste product. In the early days of getting Rubisco to work, this problem of discerning between carbon dioxide and oxygen wasn't a big deal. It was easy because there wasn't any oxygen to mess up the Rubisco. And so folks today, scientists today, point to Rubisco as an interesting, in this problem, photorespiration, as an interesting relic, it's like the appendix of photosynthesis, right? Why do you have an appendix? Maybe it was important a long time ago. It's not doing you much good today. Why do you have this inability for Rubisco to discern between carbon dioxide and oxygen? It's probably just a vestige of the circumstances under which Rubisco evolved in the first place. And so this problem of photorespiration is something that some plants have addressed head-on. There's two kinds of plants in the world. Those that have dealt with this photorespiration problem and those that are just going to make do with the inefficiency that photorespiration represents. The ones that have dealt with it, we call carbon-4 plants. The ones that haven't dealt with it are carbon-3 plants. Looks like I lost my microphone for a second. Two kinds of plants in the world. The carbon-4 plants and the carbon-3 plants. The carbon-4 plants are things like grasses and corn, okay? Pineapples. Those are carbon-4 plants. Carbon-3 plants are things like soybeans and maple trees and oak trees, things with the broader leaves as opposed to the slender, grassy leaves for the carbon-4 plants. Two kinds of plants. Those that have dealt with the photorespiration problem of Rubisco and those that have not. The way the carbon-4 plants have dealt with the photorespiration problem is they fix carbon not with Rubisco, but first with something called PEP carboxylase. They fix the carbon. They get it out of this atmospheric gas situation and put it into something that's solid, a 4-carbon sugar called oxaloacetate. And they can sequester the oxaloacetate for as long as they need. All right? When the time is right, they use that oxaloacetate to make pyruvate in carbon dioxide that can in turn be run straight through the Calvin cycle, just like the carbon-3 plants do. So this C4 pathway, this business taking place in the upper part of the slide, is the new thing, and this is sort of what every plant uses, what I've just described to you today. Carbon-4 plants have this in play. Carbon-3 plants just muddle by with just what we've talked about today. I said that the oxaloacetate can be sequestered until the time is right. You know when the right time is? The right time is when carbon dioxide levels, or better, when oxygen levels inside of the plant cells are low. When are oxygen levels low? Oxygen levels are lowest inside of a plant when they open up their pores and let the waste product oxygen gas get out. Okay? They open up those pores to let oxygen gas get out when they're not under stress for dehydration. So on hot, dry days, plant cells close up their pores and they build up oxygen gas inside of themselves so as to not let the water escape. An unfortunate consequence to the plants of saving their water is it causes them to swim around in their own waste to have a lot of this oxygen gas build up inside. In those days, hot, dry days, C4 plants can still sequester carbon. They can still fix carbon. They can still take advantage of the light reactions of photosynthesis and lock those energy gains in. The poor C3 plants have to just take it on the chin and accept that they're going to be doing a lot of photorespiration, wasteful mistaking of oxygen gas for carbon dioxide by rubisco that needs to be fixed. It comes at a cost. Some people estimate as much as 50% of a reduced efficiency in carbon fixation for C3 plants on hot, dry days compared to the energy capture that's going on in C4 plants on those same days, because the carbon is getting fixed into a 4-carbon molecule as opposed to initially into the 3-carbon molecule that you've come to know gets created by the rubisco product breaking down. All right. So, in a nutshell, here's what's happening with photosynthesis. You got your light reactions that are making ATPs and NADPHs. You're using those ATPs and NADPHs to drive the dark reactions, and you make yourself some sugars. And that's what I wanted to tell you guys about photosynthesis. On Thursday, we're going to talk about something completely different, signaling of information from one cell to another. We're getting to some serious biology. And then next week, I get to talk with you about some really fun stuff, cell division, and even a little bit about genetics. All right? So see you guys on Thursday.